Hey guys, Mr. P.
What's up, guys? It's Mr. Schmitz.
And in this video, we're going to talk specifically about cell structure and function. So diving into the cell and talking about organelle structure and function. Let's go.
For most of our discussion today, we're going to be looking at eukaryotic cells specifically. So we've got an animal cell up here on the screen for you today, and we're going to talk through the general structure and function of these cells and take a look at these organelles and what they're doing inside of the cell. Our first one that we'll start with is the nucleus. The nucleus is what contains nearly all of our cell's DNA. It's going to possess all of the instructions, which remember is what your DNA is. It's your instructions for proteins. So it's going to possess the instructions for making those proteins.
And while we talk about nucleus is a general term that is used to kind of stand for a variety of smaller structures as well. So within the nucleus, if we talk specifically the anatomy of the nucleus, there is a dense core inside where the DNA that he alluded to is going to be contained. Outside of the nucleolus, there is a two-membrane or a double membrane structure, which is very unique from a lot of other organelles. Most organelles have one membrane. This one has two, and not only does it have two or double membrane, the membrane has these large pores, which have a specific function that we'll talk about in a later date.
So moving on, connected to the nucleus is something called the endoplasmic reticulum, and we actually have two varieties of endoplasmic reticulum. The rough endoplasmic reticulum, which is called the rough endoplasmic reticulum because it is studded with ribosomes. We know that ribosomes help produce proteins, and so we know that the rough endoplasmic reticulum is going to assist in protein production. Second is the smooth endoplasmic reticulum, which is connected to the nucleus and the rough endoplasmic reticulum, but it lacks ribosomes and as a result does not help with protein production, but instead helps produce lipids.
So I like to point out the similarities of these organelles. This is essentially the same organelle. It is both endoplasmic reticulum. Notice similarities in their structure. The only difference, really, like you said, was the presence of the ribosomes on the rough and the absence of the ribosomes on the smooth.
So the next organelle to discuss is the Golgi apparatus, sometimes called the Golgi body. This is an organelle who is responsible for sort of the receiving, modifying, and shipping of our proteins and our protein products that it receives from the rough ER, which we abbreviate RER. It's going to send them out of the cell or wherever those proteins are needed to carry out their function.
And while we're talking about this, I think it's helpful to think about the cell in terms of a city using an analogy. And so I like to think of the Golgi apparatus as the post office of the cell.
I think that's a great way to think about what it does.
Mitochondria is the first of two energy organelles that we'll actually kind of highlight at the end of the video. But mitochondria is the energy organelle that is possessed by animal cells, and since this is an animal cell, it obviously is going to be included. Mitochondria is the organelle that is responsible for breaking down all of the sugar, all of the glucose, which we know is a carbohydrate, and from our previous videos, we know that carbohydrates function as short-term energy. So mitochondria are instrumental in breaking down those sugars and turning them into a usable form of energy that the cell utilizes on a daily basis, and that form is ATP. Mitochondria is going to use cell respiration in order to do that.
Well, the mitochondria is quite the powerhouse.
Some would say.
Up next, we have the ribosome, which we've already talked a little bit about in our discussion of the rough ER, but the ribosome is an organelle responsible for the production of proteins. It directly connects amino acids together to form the primary structure of a protein, which you guys have learned about previously. And this is really the driving force in protein production.
And some might look at this and say, wait, I thought you said ribosomes are attached to the rough ER. And we would say they are. However, we have two forms of ribosomes. We have ribosomes in animal cells that are attached to the rough ER, and those are primarily used for excretion of proteins, which we'll highlight in a minute. There are also free ribosomes, or ribosomes that are located in the cytoplasm, attached specifically to cytoskeleton components. Those are going to use amino acids or build proteins that stay inside the cell. So there is a difference between the ribosomes that are used on the rough ER and those that are free in the cytoplasm.
So speaking of the cytoplasm, in case you don't remember or weren't familiar with the cytoplasm refers to the area around the nucleus inside the cell, with the fluid, sort of semi-fluid within the cell, and that is where all of our organelles are suspended in and located within.
Yeah, and if we think back to previous videos and use vocab that has been attained within the class, The cytoplasm is kind of like the solvent that all of these components are kind of, I wouldn't say dissolved in, but are at least housed in.
Right.
Cell membrane is the outermost barrier between the outside of the cell and the inside of the cell, and it is composed of phospholipids. Those phospholipids are contained within a lipid bilayer, so it's 2 layers of lipids oriented heads out and tails in. Again, we've talked about the actual structure of phospholipids, and we'll highlight cell membranes in a later video, but the cell membrane serves as the boundary between inside and outside of the cell, and when doing that, regulates the movement of materials into and out of the cell. So the cell membrane is the border that kind of dictates what goes in and what goes out.
Up next, we have the lysosome. The lysosome is an organelle that aids in the breakdown of materials, so that could be the breakdown of waste, the breakdown of food, or maybe even pathogens. They do this by containing digestive enzymes. So you guys learned about enzymes in a previous unit and how they work to break apart molecules. So the lysosome is full of these enzymes, and it goes around and breaks down and breaks apart sort of all of our waste products within the cell.
So it kind of cleans up the cell.
Absolutely.
Speaking of analogy, we would think that that maybe is a trash truck.
Yep.
Garbage truck.
That would be a good one.
Yeah. Next is the centriole. So the centriole is a set of organelles. It's actually a set of 2 organelles within kind of one name, the centriole, and they are instrumental in the production of spindle fibers. And those spindle fibers that are produced during times of active cell division are going to be one of the most important things that are produced, or structures that are produced, to actually aid the cell in separating the chromosomes equally and ultimately dividing the cell. We will highlight these in our cell division chapter, but for now, it is part of the cell, and so we wanted to highlight it in the video.
You might have noticed there's a couple of organelles labeled that we haven't really discussed. Those exist within the cell, but are kind of less important for our discussion at the moment. But there are a few structures we wanted to highlight that are missing from this cell. Because this is specifically an animal cell, it doesn't have a few of these things. The first being a cell wall. So animal cells don't possess a cell wall, but plant cells do. Followed that with a chloroplast, which is another organelle that plant cells possess because they utilize photosynthesis and animals do not. And so they would have chloroplasts, but this animal cell doesn't. The final piece is our cytoskeletal components. The reason that this usually isn't illustrated in cells is because they're sort of all around within the entire inside of the cell, and it's pretty hard to illustrate that, as well as all of these other organelles, and so a lot of diagrams will leave out the cytoskeleton, but there are cytoskeletal components that make up the sort of interior structure of our cells.
And just like organisms, exo and endoskeleton organisms are going to have skeletons that regulate and control the shape and structure of the body within, the cell has a skeleton as well. There are 3 different components, and those components, like you said, would be all in here and would be kind of regulating the interior of the cell. A lot of these vesicles and peroxisomes and lysosomes are actually going to use motor proteins to move up and down these microtubules, much like a highway. And so while they're not included, it is important to note that they are there. So moving into an actual pathway that is important for our discussion of structure and function of cells, we wanted to highlight the protein secretion pathway. All of the proteins that are used inside the cell and outside of the cell are going to be produced and moved through the cell in the same way. And so the first thing that happens is the ribosomes, which are located on the surface of the rough ER, are actually going to assemble the amino acids into a primary protein structure. We've talked about primary protein structure in a previous video when we highlighted protein structure. Remember, primary proteins are just a single chain of amino acids. Once they are assembled, they're going to be immediately moved via a vesicle to the Golgi apparatus, which is in very close proximity to the rough ER. And before we move into that, it's important to note just how much rough ER a cell that is in charge of protein secretion will have. Why do you assume that a cell in charge of protein secretion would have so much rough ER?
Well, we established on the last slide the function of the rough ER is to help produce proteins. And so certain cells that are responsible for the production of vast amounts of proteins would need much more rough ER in order to keep up with the demand for those proteins.
Right. I mean, this cell, I know, is a diagram and is a artistic rendition of a cell, but this cell has probably 50% of its interior volume filled up with ribosome-rich rough ER. And so this is obviously a cell that's probably going to be utilized within a protein secretion pathway. From there, the proteins, which are going to be kind of represented by these little dots, which you see being secreted ultimately at the cell membrane, are going to move through the Golgi apparatus. And while we're talking about rough ER and Golgi, the rough ER is a set of interconnected flat membranes. All of these membranes would be connected into one continuous membrane. The Golgi apparatus is a set of non-interconnected membranes. So this little structure or this little section of the Golgi is going to be completely independent of this section, which is completely independent of this section. So that's just one of the many differences between the two. But these proteins are going to be moved through the set of flattened sacs that make up the Golgi apparatus and will be chemically modified and folded and finished into the tertiary and quaternary form of protein structure, which is taking on that three-dimensional shape that will be a finished product.
So ideally, when they leave the Golgi, they have completed their folding and modification, so now they would be a functional protein is what we would call that.
Right.
The final step is once that Golgi packages those, the proteins are able to be sent through the secretory vesicles for release from the cell. And so that's illustrated here at the end, like Mr. Pfeiffer was illustrating just a minute ago. The little dots being our proteins as they are released from the cell through those vesicles and transported out of the cell extracellularly to help with whatever function they have been produced for.
Yeah, it is important to note that not all proteins that are made within a cell are going to be used within the cell. Some of the cells that make up the tissues in you as an organism are used exclusively for the production of extracellular metabolic proteins. Think about the pancreas. The pancreas is basically a secretory organ that is producing proteins and hormones that are proteins that are going to be moved into the stomach and moved into the small intestine that help you digest food as an organism and then assimilate the energy that you get from the food.
To wrap up our discussion of cell structure and function, we wanted to highlight two specific organelles. We talked about each just briefly on that first slide, but these are what we would call our energy organelles. So pictured here you have the mitochondria and the chloroplast. And like Mr. Pfeiffer said about the mitochondria, it is responsible for carrying out cellular respiration, which is carried out by both plants and animals. So both plant and animal cells possess mitochondria, whereas the chloroplast carries out photosynthesis, which is only done by plant cells. So we're going to look at the structure of these two organelles and talk about how the structure contributes to their ability to do these processes.
Yeah, one of the key things about the energy organelles is that there are two membranes in both. So you'll notice that in both there is an outer membrane and an inner membrane, and then there is a space between the two membranes. The two membranes are not right on top of each other. In the mitochondria, we have an outer membrane and an inner membrane. We call the inner membrane folds. You'll notice that it's all folded up. The cristae. The purpose of the folds, as we'll get into more intricately when we get into cell respiration, is to increase surface area, which obviously increases efficiency. In the chloroplast, we have an outer and inner membrane, but the inner membrane is not folded up, and instead we have these really flat discs, which, again, help to increase surface area, but these are kind of like solar panels when they are utilized to absorb sunlight.
Right, so in the chloroplast, sunlight is reaching each of those discs, and that is where one of the steps of photosynthesis is occurring. And so the more of these discs that you have, the more, which are called thylakoids, the more of these thylakoids that you have, the more photosynthesis each chloroplast can generate.
Right, so chloroplasts, plant only, absorb sunlight, utilizing these discs called thylakoids to produce usable energy for the plant cell. Mitochondria is found in both plant and animal cells. Basically, all eukaryotic cells that are utilizing their food for energy have to employ mitochondria to produce ATP, and they do that basically through a series of metabolic reactions, most of it occurring at the cristae or the inner membrane. Again, we're going to talk about more of the cellular energetics in later chapters, but this was just a highlight of structure and function. These organelles, just like all of them, have a specific structure, and that structure makes it very conducive to do the function that that particular organelle utilizes.
Right. So, as you've heard before and as you will continue to hear throughout the entirety of this course, structure dictates function.
And that's all we got for you today. Thanks for watching.
Thanks so much for being here.
Bye.